So, I'm met today by Katie Moran, who is a fantastic graduate student at the University of Manchester. And today we're going to talk about a great number of things, including dementia, hopefully, and her various outreach methods, and a little bit about your history, too. So, the first thing that I wanted to talk about is what is it that you want to work on? What is it that you work on as a graduate student? What research are you doing? And hopefully, what purpose should it serve?
Yeah, so my kind of research focus is I'm interested in the relationship between cerebrovascular risk factors, how they contribute to cognitive decline, and how kind of changes to the brain structure kind of mediate that relationship. And the reason why I focus on this is because vascular risk factors are actually linked to dementia later in life,
Mm,
and they're actually modifiable. So risk factors can include things like high BMI, high waist to hip ratio, diabetes, cholesterol. Not all of them are modifiable, but a large proportion of them are. So if we target them early enough, we might be able to prevent a large proportion of dementia diagnoses.
yeah,
So I'm basically interested in this relationship from a much earlier stage. So I'm looking at kind of mid age, around 45 to 60. And I use large scale data sets like the UK Biobank to kind of study this relationship.
that's perfect. So I was wondering about the ways by which your research can be applied to dementia in particular, because I found that to be really interesting. I know that you, for instance, were considering doing work on a dysconnectome,
Mhm.
which is, if I'm not wrong, it's work to understand how the vascular changes in someone's brain can affect their later chances of dementia. Am I right?
So, the reason why it's so critical to kind of look at the vascular implications of dementia is because the white matter in the brain is particularly vulnerable to poor vascular health. So, the white matter has less collateral circulation than grey matter. So,
Hmm.
when things go wrong, it doesn't have as much backup as grey matter does.
Mm.
And also, the kind of cellular population within white matter is very different to grey matter
Mm.
in terms of when there's a hypoxic environment, so when there's low vascular innervation, the cells react quite badly to it. So the main contender within the white matter is called a oligodendrocyte,
Yes.
and that basically coats the lining of the neurons within your brain. And white matter is white because it's fatty, and oligodendrocytes make this fatty sheath. We
Mm.
call it myelin. And when there's a hypoxic environment, this myelin begins to kind of unravel and degenerate.
Mm.
And that's when you start to see these white matter lesions. And
Yeah.
that's what I'm interested in. And the disconnectome that you just mentioned is looking at these white matter lesions and assessing how that affects the connectivity of the rest of the brain.
Hmm.
So traditionally, we would just look at the where the region of the lesion is. So let's say it's you've got a lesion in the occipital lobe. How is cognition affected just based on that? But the approach that I'm taking is if you've got a lesion in the occipital lobe, what tracks does that affect? And then how does that better reflect cognitive change if we consider the whole disconnectome rather than just a lesion alone?
Mm. Oh, so if I'm understanding you correctly, it's not so much, where is the cut, so to speak,
Yeah.
in the brain, but rather where is the collective source of this cut projecting towards,
Yeah,
right?
exactly. What are the downstream effects of this singular lesion?
Hmm.
Because the brain operates as a network, so it seems really reductionist to just consider it as just a single lesion.
Yeah.
We should really be considering how the wider network is impacted by just this one lesion.
Yeah, that makes sense. And so, in your work, what do you think? What kinds of implications could this have? So, for example, applications. What could people do with that knowledge?
Yeah, so as I was saying, vascular risk factors have this really close relationship with dementia later in life, and even just cognitive dysfunction. So if we try and assess this relationship and identify a key window in which alleviation of these risk factors benefits later life cognition and benefits our white matter into older age, then that would be a really great step forward to preventing dementia.
Oh, yeah.
um, it's likely that there's going to be this kind of limited window in which, when we alleviate these risk factors, there is actually going to be a benefit. So, identifying when that window is in
Mm,
terms of age and in terms of risk factor proliferation,
yeah.
that could be really helpful for dementia diagnoses.
Wow. So in that case, and tell me if I'm getting you, you could add another method of understanding how does dementia come about and other age-related diseases like it of the brain. So that seems to be a way to pinpoint better what could be the actual causes of these diseases. Is
Yeah,
that right?
exactly that. And it's really only in recent years that this kind of vascular component of dementia has been considered.
Mm,
Prior to that, it was just thought that neurons degenerate for some reason. We don't really know why. Perhaps tau plaques and amyloid beta, which is definitely still the case, but there are these risk factors that happen much earlier on that we could perhaps prevent in order to prevent a later life dementia diagnosis.
yeah, I heard that plaques, that basically means that different parts of your brain start to clump together, if I'm not wrong?
Yeah, exactly that. So these plaques occur in our brain naturally. We all have them. We will both have them. But in people who have dementia, they're just not cleared as well, and they
Mm,
begin to clump, and then they become much more difficult to clear. But that's at the point where you will really start to see cognitive change. The point that I'm interested in is much earlier on, so we can kind of do things to prevent that happening later
I
in life.
see, I see. So then the plaques are like cognitive scarring almost,
Exactly.
whereas what you're trying to suggest is what causes that, what is producing these things, and how can we prevent that? So then to prevent the
Exactly.
scarring as well. That makes sense. And and so in your case, I believe that you've been doing research related to that, and right now you've been writing a paper which was trying to understand that a bit better. So could you explain that to me a little bit?
Yeah, so I work with a large scale data set for called the UK Biobank. And if anybody is unfamiliar with what the UK Biobank is,
Mm.
it's a humongous cohort of 500,000 people
Hmm.
which have been collected around the UK. I think there are a couple of sites in Manchester, there's one in Newcastle, Glasgow, and then a couple down in Oxford and Cambridge.
Yeah.
And so, yeah, they've sampled 500,000 people, and at baseline, cognitive measures and health data was collected. On the second visit, they collect health data again, some cognitive measures, and they do a full body image.
Yes.
And then they have a second visit where they do more cognitive tests, and they've just started repeat imaging,
Mm.
which is great because you have longitudinal data. So at the minute, I have just analysed this data in terms of cognition, in terms of health information, and then in terms of the imaging data, specifically brain imaging. And I was essentially interested in the relationship between cerebrovascular burden and processing speed. So, processing speed is thought to be a cognitive domain that precedes dysfunction in other cognitive domains.
Mhm.
If you kind of think about it, you can consider processing speed to underpin quite a lot of other cognitive domains. So,
Yeah.
for example, working memory.
Yeah.
If I give you a sum to add up, then you have to do it quickly or else you forget the sum.
Yes.
So a lot of these domains rely on processing speed. So I targeted that as a potential early neurocognitive marker of
Yeah.
cerebrovascular burden. Um, and then I was also interested in whether white matter kind of mediates that relationship. As
Mm.
I've already mentioned, white matter is particularly vulnerable to poor vascular health. So I was interested in whether white matter mediates that relationship. And I was interested in kind of establishing the regional specificity of this effect. So
Yeah.
is there a specific region that mediates it? And then also the cognitive specificity. So is this relationship unique to processing speed?
Mhm.
And essentially, what we found was that the effect of cerebrovascular burden on white matter is unique to anterior white matter integrity.
Mhm.
And there's a couple of reasons for this. One could be that there's this kind of first in, last out hypothesis. So the anterior portion of your brain is the last to develop. So it could be
Mhm.
just particularly vulnerable for that reason alone. Another reason is that there's slight differences in vascular innervation. So the frontal lobe has slightly less vascular innervation. So it could be that when things go wrong, it's the first to go again.
Okay.
So
Yeah.
that's why it could be vulnerable to vascular risk factors.
And when you mean vascular innovation, what exactly do you mean by that?
So that could mean the amount of circulation that happens in that region, the
Mhm.
amount of blood vessels or capillaries in that region.
Mm.
There's just slightly less innovation, if that makes
That
sense.
makes sense. Yeah.
And then in establishing cognitive specificity, we found that cerebrovascular burden was associated with slowed processing speed. And we also found that cerebrovascular burden was associated with poorer cognitive performance.
Yeah.
However, for cognitive performance, this wasn't mediated by anterior white matter integrity. So it could be that there's a more specific region within the anterior lobes that are responsible for this change in cognitive performance, or
Mhm.
it could be that there's a different region of the brain. So that's where the disconnectome comes in to look at how
Okay.
more advanced white matter lesions can kind of explain that, really.
Mm. Mm. I understand. So in that case, if I'm getting your research correctly, first of all, you're trying to understand what effects does it have on the frontal part of your brain,
Yeah.
which is corresponded to a lot of the most human qualities that we associate with people. So, like complex processing, etc. And then what you found is that the and tell me if I'm getting this right, that the processing speed is somewhat related to then succeeding dementia cases,
Mhm.
but it's not fully. So there's something more to that picture.
So in the baseline model, we're interested in establishing this effect between cerebrovascular burden and processing speed. And what we found was that increasing cerebrovascular burden, so if people have more of these risk factors,
Yeah.
that's associated with slowed processing speed. And this effect is mediated by this change in anterior white matter integrity.
Hmm.
So that was the kind of baseline model, and that was what we was hoping to establish, and it's what we did establish.
Hmm.
And then in the second model, we wanted to establish regional specificity. So we swapped this anterior white matter integrity variable out for posterior, and we found that this effect didn't really work as well. The model
I
didn't
understand.
fit. The pathways were no longer significant. And then for the third and final model, we swapped the outcome measure, which was processing speed, out for cognitive performance to see what the model did then. And the model fit just as well, but the mediatory effect of anterior white matter integrity didn't fit the model or was no longer significant,
Okay.
which suggests that cerebrovascular burden affects more than one cognitive domain, but this domain is better explained by a different region of the brain, and that's why we're doing the
Mm,
disconnecting.
I understand. So if I'm getting
Yeah.
you, you found that certain parts of the brain are affected by vascular burden,
Mhm.
but not all of them, or at least not all of them to the extent that you would naturally suspect.
Mhm.
So, okay, I think I understand that now more. Yeah. Um, yeah, yeah. And so what I'm curious about is that in particular, how did you arrive at this burden being a possible explanation for why later on these diseases can come about?
Yeah, so I think I kind of started my research interested in vascular health anyway. So my master's project kind of looked at how um, when your brain becomes active, how does the hemodynamic response change as you age? So I already kind of had that interest in vascular health.
Mm-hmm.
And then when I came here, my supervisors were kind of like, "What are you interested in?" And I said vascular health. And then falling into a cognitive lab, it just kind of felt like the most natural step to do.
Mm-hmm.
And then having read through the literature, I realized that there's this kind of gap
Mm.
for modifiable risk factors for dementia. And that's where vascular health just kind of slotted in there really nicely.
Mm-hmm. So it just seemed to naturally click in
Exactly.
that possible explanation for these later cognitive challenges that people have.
Yeah.
Okay, that makes complete sense. And now, but this research that you're doing is not the only thing that you're focusing on because you're also, in addition to the neuroscience that you're doing, you're also teaching. And not only that, you're also trying to do outreach
Mhm.
projects. So I was very interested in learning about what it is exactly that you do there. So let's start off with your teaching. What is it exactly that uh, you occupy yourself with outside of science itself?
Yeah, so my PhD is a little bit of a package deal. So I do part-time PhD and part-time teaching as a GTA.
Hmm.
And that's roughly split about 50-50. And I teach on a whole range of modules, seminars, tutorials, workshops, and then
Hmm.
I do marking towards the end of the semester as well. As well as the both of these, I do quite a lot of outreach work, as you mentioned, and that kind of takes different forms.
Mm.
So I'm part of the Manchester Neuroimaging Outreach Group, and we do quite a lot of neuroimaging outreach at primary schools and at high schools.
Hmm. How does that
Um,
look like?
so it really varies dependent on the school you go to. We did quite a lot of this during COVID, which involved either giving presentations or making materials to send out to schools that they can show their pupils.
Yeah.
Um, Myself and another GTA called Rowan, we developed this neuroimaging video to explain how fMRI works to a lay audience,
Hmm.
and we started kind of working on a series of those, which was really cool. I'm also a widening participation fellow, which means I go to different primary schools every year. I've been to one for the past 2 years now, and I'm going back again this month.
Yeah.
And that's to a group of year ones. And we basically talk about the brain and what different regions of the brain do.
Yeah.
And we play a really fun game called Guess What the Fruit Is in the MRI scanner, where I show them MRI scans of different types of fruit and they have to try and guess it.
Yeah.
And it's just like a really fun, rewarding session. It's definitely very different to teaching undergraduate students, year ones, undergraduate students, very different,
Yeah.
but really interesting.
We're more boring.
Just less inclined to give answers. Year ones are very keen.
Really?
Yeah.
Oh, well, that's great. They already know what an apple will look like on an MRI scan.
I got a question from a year one once that was, "Why does my brain tell me to pick my nose?" And I've not had that from an undergraduate yet.
No, 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 hopefully not. Yeah, that totally makes sense. Um, so I'm really curious about what exactly do you find to be the differences between undergraduate students and younger audiences? Because I know that, of course, there's are some differences. There's more of a maybe, hopefully, a more mature approach to the science itself. But I'm sure that there's some commonalities amongst them. There's this maybe curiosity or drive to learn more about a subject that they find really interesting.
Mhm. Yeah, I think you've hit the nail on the head. I think there are quite a lot of similarities between year ones and undergraduate students, like in terms of the curiosity and their passion for the science.
Mm,
Except with year ones, it's fueled by the fact that they don't know anything about the brain, whereas when you get to being an undergraduate, you're fueled because you know so much about the brain and you want to know more. So slightly different motivations, but definitely some similarities between the 2.
that's I definitely found that to be the case myself.
Mhm.
I even remember trying to explain to my little cousin a little bit about the brain and a little bit about psychology as well, and I was laboring over trying to make a complex point, but that completely wasn't working. So I had to go for a more simple but more relatable approach.
Yeah.
So it was like, when you meet someone and you feel really sad, yeah, that kind of stuff.
Yeah, I've
So
definitely found the same with year ones. I feel like it's really helped me to develop my teaching style because you're really challenged to explain these really complex topics
yeah.
to a group of students who don't know what cells are, they don't know even know whereabouts the brain is in the body. So it's really starting from the beginning and breaking everything down as much as you can. And then I felt coming back to teaching, I was like, oh, okay, this is a lot easier now.
Yeah, yeah, yeah. That has to be a skill that you really develop through talking to people who completely. It's as though you're speaking another language. It's
Exactly.
when you start talking about neurons and cells and all that. That has to be something that they cannot relate to yet because they haven't had that experience. So you need to break it down into okay, what is a cell? What is a neuron?
Yeah,
If I'm not wrong.
it's so funny, explaining it because they just can't believe that this world exists inside our body. It almost seems like witchcraft to them.
Yeah, yeah. It, it is a bit like witchcraft, to be honest, in my view. Um, so now I was a bit curious as to you said that you do teaching and you do a
Mhm.
bit of outreach. And so I'm curious as to where the motivation for doing things like that comes from.
Yeah, so I myself come from a winding participation background. I came from a low income household, and I was from a very small group of people from my high school who went to university. The majority didn't. They
Mm,
stayed back at home to do apprenticeships, or they went straight into jobs.
yeah,
So I was kind of heading into the unknown a little bit. No one from my immediate family had been to university either. So yeah, I was kind of heading into this unknown, and it took a university to come to my high school and kind of inspire me to go on and to go to university, or
mm,
else I think I might have been led down a very different path. Um, so I was inspired by that university trip or them coming to my high school. So I really feel like it's necessary for me to do the same and inspire other students who might not be considering uni, but they would thrive here.
yeah.
And I think it's so important because there are so many barriers, and I think breaking down those barriers means people will be able to access university much more easily. Um, yeah.
Yeah,
Mhm.
that sounds very very sweet. Um, one thing that came to my mind when you said that is well, of course, you'd be interested in doing that because you can actually appreciate the value that it provides someone because you yourself were affected by it. So I can definitely understand it in that context. And so I'm curious, what do you find to be the biggest barriers as far as you see them for students who may come from lower income backgrounds for going to university?
Yeah, I think there's a whole host of things. I think for me personally, because there weren't many of my peers applying to university, and obviously my parents and my brother hadn't applied to university either. Nobody really knew what the script was. Like, how do I write a personal statement? How do I apply to university? How do I pick which one I want to go to? How do I know which one is a good one? So I kind of just went with the flow, and but I think that was a really tricky bit, kind of putting your foot into the unknown.
Yeah.
But obviously, I'm very glad that I took that leap.
Yeah, I'm sure that a lot of students at the University of Manchester in particular, because it's such a global
Yeah.
university, can appreciate what you're saying as well. It's just like you feel a little bit out of place because you're maybe coming from a completely different system, let's say from China or Poland, like myself. And it seems like just a completely different experience, the application process and all that. So, yeah, I can definitely um, appreciate what you're saying. So, it sounds like one of the big difficulties is just having that sense of community or sense of understanding
Mhm.
from others, from your peers, who going to university, and for that to be a viable or maybe a easy option. So that seems to be something you found to be the case. And do you believe that through the outreach that you're doing, you're giving students the chance to feel that they have more of a community as well?
I like to think so, and I think just inspiring students to think beyond the application process and really consider what university life is going to be like
Right.
and the type of things that you could learn and how exciting the world of science is. I think that's really what I try to carry through outreach work.
Yeah. 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 Of course. Well, that's what we're hoping to do here as well, right? So that definitely makes sense to me. Now, one thing that I've also heard is that in addition to the teaching and outreach you do, you're also focused around trying to improve the administration or the staff experience here at Manchester as well. So I was curious to understand what you've been working on in that area because I heard that you've been working on a peer review
Mhm.
system with other graduate students and a professor, if I'm not
Mhm.
wrong.
Yeah, so myself, Meg Shepherd, Lucy Hume, and Lizzie Lewis have all been working to develop what we call a GCA peer review process. We're not really reinventing the wheel or anything like that. It just comes from a place that GCAs are given their initial training and then they're kind of let out into the wild and they are offered support as they teach, but there's no real follow up or any additional feedback given or anything like that. So we really wanted to give GCAs an opportunity to have some feedback.
Mm,
Um, Not only to just increase their confidence, but by providing them with evidence of their teaching so that they can apply for teaching qualifications called the Advanced HE,
okay.
where you need to hit certain targets in order to be able to apply for it. And by us doing this peer review, we can say, ah, you've done this in this session, or you've demonstrated this in this session, and that helps to give them evidence to apply for it as well.
That makes sense. So if I'm getting it, it's as though you're almost making a scientific approach to this. You're trying to systematize it much more than it was before. And there's also the element of observation of people, so they're getting the chance to see how they're improving and what they're doing right, what they're not doing right. And so to me, that's just like from a personal standpoint. I've worked as a student representative, and students have said repeatedly that that's something that they wanted. So I can definitely um, attest to it
Mhm.
being a very valuable um, activity. Now, I'm curious as to one more thing, and that's when you're thinking about your PhD work in general and hoping to do something within that area. What's your dream here? What are you hoping to achieve through this?
That's such a tricky question, and I think if I had unlimited funding,
Mm,
even to be fair, even if I had unlimited funding, I think it would be really difficult to match anything close to the biobank because
yeah.
they have such a rich data set and it's so high quality. So I think even if I had all the funding in the world, I would love to just continue with my biobank work because they, like I said, it's just such rich data. It's so well collected. It's really well standardized, and again, it feels like there's such a big community around biobank. There's so many people you can reach out to and say, "Hey, I'm working on a biobank project. What are you doing with your data or how have you analyzed this?" And there's a whole forum of users that chat to each other and talk about different analyses and things like that. So I'm really a biobank hype gal.
Oh yeah, yeah. But from what I'm getting and from what I got from Megan as well, who was my previous interviewee, this is like a almost revolutionary new way of deriving new scientific knowledge. This is just a giant, giant undertaking, which seems to be at the scale of like some of the cancer research projects which are happening all over the world now. So that seems to be has to feel really great to feel like you're on the cutting edge of something really, really significant like that.
Yeah, absolutely, and it's cool going to conferences because I'll always put biobank on my poster because obviously that's what all my work has been about up to now, and people always come over and ask questions like, "How have you accessed the biobank? How have you found it? Do you have any feedback for the biobank?" And my supervisor, Nils, is really involved in the biobank process at the minute, and he's the lead for our school in the biobank research group. So it's really nice to be involved in that community, and like you say, it's so humongous,
Yeah,
and really bigger than any other cohort out there. So it is really amazing to be a part of it.
yeah, yeah, definitely. Now, one more thing with that, which I was wondering about, was when you're thinking about your work, let's say 10 or 20 years from now, what do you think, maybe your field in general, what do you think are the new innovations that are going to happen here? What do you think are going to be the new um, possible areas of exploration for people who might want to get into the same field as yourself?
I think there's a couple of different angles you could take it from. So I think what I do now is kind of at a macro scale. I look at lesions, macro scale biomarkers within the brain, and vascular risk factors. But I think you could even shrink that down to micro scale. So at the level of the neurovascular unit, there's so much interesting research going on. The lab that I worked in when I was doing my master's in Sheffield,
Hmm.
they kind of focused on more a cellular approach to the neurovascular unit. And essentially, what happens is when your brain becomes active, there's a kind of cellular cascade from the neurons down the astrocytes, which then reach the blood vessels. And that notifies the blood vessels that the neurons are active and they're going to need more blood.
Mm.
So the astrocytes then dilate the nearby vessels to supply the neuron with blood. But there's so many recent discoveries at the level of the neurovascular unit. I think there was a new cell called a caveoli found recently that's recognized to kind of help with this dilation process.
Hmm.
So I think that is such an interesting aspect to take. And I think, yeah, in the next few years, there's going to be so much more interesting research going on there.
Wow, so that seems really interesting um, that vascular unit within the brain, because I've never really imagined the brain as almost like a muscular system,
Yeah.
because it seems from what you said that as you need more to exert a bit more by using the neurons, there's more blood going into the areas where
Mhm.
they're working. So it seems like it could be very much paralleled by how the muscles work in your body as well.
Yeah, so neurons have very little immediate reserve. So as soon as they become active, they use that up almost immediately. So
Mhm.
they really need to rely on this efficient delivery of nutrients, which is facilitated by lots of amazing cells like astrocytes and smooth muscle cells that help to dilate nearby blood vessels and get those nutrients to the neurons as they need it. So it's a really interesting process and mechanism. And there was some work that came out recently that kind of looked at these downstream effects. It's thought that astrocytes and smooth muscles can kind of signal downstream to dilate even larger vessels at the surface of the brain. So
Okay.
it's not just going on locally, it's kind of a network based approach for this kind of vascular unit, which is really interesting.
Yeah, yeah, that makes sense. Never really thought about it that way, but that makes complete sense. And from what you've said, I can also pick up that that's why the brain is so fragile because you stop it from receiving blood for 2 minutes, and then it dies.
Mhm.
So that kind of gives a sense of why things like strokes can be so harmful. Is that right?
Yeah, absolutely. We've kind of touched upon, there are definitely regions that are more sensitive to blockages or hemorrhages. Most regions kind of have collateral circulation, which means there will still be blood supplied for a certain amount of time, but it can't last forever. So there definitely needs to be medical intervention as soon as possible.
Yeah.
But the vascular system is so interesting. I feel like it's overlooked too much.
Yeah, yeah, definitely. One thing that I learned about um, the brain's vascular system is that it's as though there's almost two different interconnected systems. So there's the ventricular
Yeah.
system and then also the vascular one.
Mhm.
So is there any relationships? Of course, there are to some extent, but are in your research itself, have you considered both of them and how they integrate together? Or have you focused more on the bloodstream itself?
So I don't necessarily look at cells within my research because that's just not a skill set that I have.
Hmm.
And with my focus on the biobank, I tend to focus on the whole brain or
Yeah,
kind of connections within the brain. But the way that the kind of vascular and ventricular system works is that the vascular system comes in, supplies these neurons with the oxygen that they need, and then the blood supply is then given the leftovers, so the
yeah,
deoxyhemoglobin,
yeah,
and then that's transferred into the ventricular system, which is then drained out of the brain and then into the body to then be
yeah.
recycled for something else.
So it's almost like a sewage system,
Exactly
a
that.
water filtration and sewage system.
Yeah.
So that's perfect. That's exactly how I thought about it when I
Yeah.
was learning about it. It seemed very much like a city's toilet.
Yeah.
So so that's one way to see it.
Mhm.
Um, and so I'm also curious because right now, I suppose you've finally got the chance to understand a bit about your field because as you move from being a master to a PhD student, you finally get to understand your field from a more holistic perspective,
Mhm.
if I'm not wrong. Um, and so, have you found what have you found to be the most interesting in transitioning into that kind of um, different state of working?
Yeah, so I think when I was an undergraduate, I felt very independent, especially when I was doing my dissertation in my third year. But looking back, I realized it was heavily fed by my supervisor, which of course is what's expected during undergrad. Um, but that kind of experience really shaped my interest in going into neuroscience. Um, because my project used a method called uh, high definition transcranial direct current stimulation to see whether if we stimulate the insula, can that change our perception of itch.
Hmm.
So that was really, really interesting for me, and that kind of spurred me on to go on and do research. And then when I got to my masters, I was definitely a little more independent and kind of got to shape my project a little bit more on my own,
Yeah.
but then still heavily guided by my supervisor. And then since joining my PhD, you're really thrown in at the deep end, and it's really scary at first because you kind of think, I'm not old enough to be making these decisions about my research. I need lots and lots of input, which of course I still have from my supervisors. But the PhD is a much more independent process, and you're expected to kind of come up with your own ideas and or present them to your supervisors and ask for their input. But your research is really shaped by you and what you know and what you've read, rather than being heavily shaped by your supervisors.
Right, right. Yeah, so it seems like you're as you go along, you're slowly given more authority or
Yeah,
autonomy over your activities until one day, I suppose, you're going to do it all by yourself and you're going to be supervising some other
which is
people.
terribly scary.
Terribly scary. Yeah, it seems to be. And with that in mind, I think that some people who are interested in doing PhDs themselves, or at least interested enough in the process by which people become scientists. Um, is there any advice or suggestions that you'd give, given your experience with that?
Yeah, I think just being so just to kind of take it back. So when I first came to uni, I really wanted to become a forensic psychologist.
Hmm,
That was the dream. I was really interested in serial killers and murderers and all the good
who
stuff,
isn't?
naturally. But then when I got to university, I did some volunteering with the police as what's called an appropriate adult, where you go and you're on call and you get called out to the custody suite when an adult with either learning difficulties or if they have mental health issues, you kind of sit with them during their caution and during their interview to ensure their rights are being upheld,
Yes.
and the police aren't kind of crossing any boundaries and forcing a false confession. Which was a really, really interesting volunteering opportunity. But I quickly realized that it wasn't for me to
Hmm,
become a forensic psychologist. I thought the environment was a little bit scary and I just didn't enjoy it. So after that, I was at a little bit of a loss as to what I'm going to do now.
yeah.
So I took on a bunch of different volunteering opportunities, one of them being a dementia friend, where I kind of met with a group of people who had dementia every week. And every week I'd introduce myself for the first time to the same group of people who would never remember my name. Um, and yeah, they'd never remember my name, and we'd always have a kind of cup of tea, and they'd retell the same stories, even though I've heard them 100 times before. And it was kind of that point where I was thinking, why do these people have dementia? What makes them different to my grandparents who are of a similar age but cognitively healthy?
Hmm.
So that kind of spurred me in the direction of dementia. And then, as I mentioned, these research projects that I did got me into neuroscience, and that's where it kind of crossed over. But I think the advice that I would give to undergraduates is take on volunteering opportunities or as many opportunities as you can to try and figure out what it is you want to do. So
Yeah.
I thought I knew what I wanted to do, but of course I changed my mind. So if you're in the same position I was, try and take on these opportunities. There are so many amazing charities and amazing opportunities that you can take on to try and figure out what it is you want to do. So Dementia Friend could be one of them.
Yeah.
There's lots of brain injury charities that people can work with. There's lots of homeless charities. Um, that will all offer amazing experience to try and figure out what it is you want to do.
Hmm. And so, in the area of Manchester, and I suppose relevantly for University of Manchester students in particular, where do you recommend getting started? How would you go about beginning to volunteer?
So it really depends on what it is that you want to do, but there's quite a lot of national schemes. So the appropriate adult scheme with the police that I mentioned,
Mhm.
that's a national scheme. So there should be a base available in Manchester. There's a brain injury charity called Headway that are always accepting volunteers, which is really amazing. And then even within the university, I know there's quite a comprehensive list on the volunteering page on the university the SU website.
Yeah,
So I'm sure there'll be lots and lots of opportunities there.
for sure, for sure. I personally looked through a few of these websites, and I can attest to that for sure. Um, So, pivoting slightly, I was curious to understand when you do science by yourself, because I think that there's a personal component to whenever someone does research. It's not only because you want to derive some utility for people, but there's also a bit of a personal element where you there's some experience that you get out of it which you just cannot get out of anything else.
Mhm.
So for you, what would you say that's like the driving feeling that you get from doing something innovative or doing something with other people which can actually bring value?
Yeah, so kind of like I mentioned,
Hmm.
a large motivator was considering why these people I volunteer with every week can never remember my name, but for some reason my granddad does. My
Mm.
granddad does even. So that was a really big motivator. But I think I am just a natural problem solver. Like I really like a challenge. I really like to solve problems. And with the specific PhD I do, it really relies on some doing some coding, which I didn't have a lot of experience before my PhD.
Hmm.
But now I use a computational modeling approach towards my research, so it's something that I rely on heavily now.
Hmm.
Um,
And computational modeling meaning.
so I build models using software such as Python or R
Hmm.
that can kind of represent the data. So the
Visualize it, yeah.
yeah, exactly that. And then we can statistically compare the models to see which best fits the data. We can build predictive models. So that's the approach that I kind of use to my research. And it's funny because when I was an undergrad, statistics was my worst subject. I absolutely hated statistics.
Uh,
And now it's my favorite part about my research. I love doing the analysis, love making pretty graphs and nice brains and
hmm.
stuff like that. And I also love teaching stats to undergrads, especially second year,
Yeah,
because I feel like you really see that light bulb moment because students come in exactly as I did with this huge sense of anxiety towards statistics.
yeah.
And being able to help them past that and to see those light bulb moments is really amazing.
Yeah,
Yeah.
yeah, that sounds like something that could really fuel your drive to continue working with students in
Yeah.
that way. Just that sense of, oh, I got it now. Yeah, I can definitely attest to statistics being that way for myself and for many other students I know, which is just it's very challenging, but once it's clicked in, it just
Yeah.
stays there, and it seems like now it makes sense. And what's fun for me, especially, is recently because I finally got to understand statistics a bit more. When I look at a research paper, which often uses a lot of statistical methods, which I've learned about, I actually understand what the things mean. It's like, oh, I know what a P is.
Yeah.
So to me, it's a really empowering feeling to get um, to almost be able to understand that language of scientific research, which before that point I've tried to do, but it was always so challenging with all the foreign statistical names which no one really knows. ANOVA, forget about it.
And that's exactly how you feel when you run your own statistical analysis using computational modeling or using the UK Biobank. You get the output, and you're like, oh, this is so overwhelming. I don't know what I'm doing. And then once you have a read through and figure out what things mean, you have that kind of moment, that light bulb moment,
Mm.
exactly like you do with stats, which is amazing.
Yeah, yeah. It seems like what you're doing with the statistical modeling, and something that I believe seems to be paralleled in other disciplines, is you have this just a lot of raw data which seems to be unconnected in any way, and that's what seems overwhelming. And then slowly you start to find the patterns amongst these things and try to unify them under some common explanation.
Exactly that.
Yeah, so like in your case, um, trying to understand the vascular system and how it could be the explanatory variable
Mhm,
within the brain. So that seems like something that's really core to what we do as people who enjoy doing science,
mhm,
um, and something that I really enjoy about that subject as well, or field. Um, so one more thing that I wanted to ask you, it's when you personally see yourself in 10 years or maybe 5 years, depending on how long you want to look ahead, um, what do you think you'd like to be doing most?
It's a really tough question. So I think when PhD students are faced with the graduation finally, and they've finished their PhD, you really have two options, and the two options are to either stay in academia, whether that's in research or whether you take on a lectureship. If you were staying in research, you would do what's called a postdoc, a postdoctoral position,
Mhm.
or you can go into industry and work for pharmaceutical companies or other kind of big industry companies.
Yeah,
And it's really difficult to know which road to take, because if you take a postdoc position, quite often they're short-term contracts, so that's normally either 2 or 3 years.
yeah,
So there isn't a great deal of stability that comes with postdoctoral positions, but it's an opportunity to kind of diversify your skill set that you've learned during your PhD.
yeah.
And quite often they're taken abroad. So a lot of PhD students, or after they've graduated, even take postdoc opportunities in Canada or the US or elsewhere in Europe, just to kind of meet other labs, diversify their skill set. Um, some people stay to do lectureships, so just stay on and do teaching,
Mhm.
and then but I feel like I find industry really appealing,
Mm.
just for the stability component. But also it's kind of the unknown. I spoke earlier about the unknown being really scary, but now I find it really interesting.
Yeah,
So I think ideally I'd like to do a couple of years as a postdoc abroad somewhere in a nice sunny country that isn't rainy Manchester.
yeah.
And
Like
then
whales.
yeah, exactly, Wales. And then perhaps settle down in an industry job after that. I find medical liaison positions really interesting, and that's essentially where you translate science from pharmaceutical companies to the public or to doctors or whoever's going to be using the drug that they've developed.
Mhm.
I find that science translation positions are really interesting, so I'd love to do something like that.
Mm, that's really interesting. And my understanding of um, the being in industry as a scientist is that because you're working with some pharmaceutical company, you almost never have the difficulty of not enough resources. Or
Hmm.
if you do so much less often than just by being a, let's say, postdoctoral student. So, do you think that that's one motivator for doing something like this? Just the sense of I have the ability to do much more within that area than I would do just as a postdoc.
It's very tricky, and I feel like even now I can feel myself swinging back to becoming a postdoc,
Yeah,
because in industry, while you essentially have unlimited money, you don't necessarily have unlimited options.
yeah,
Because they've spent so much money developing this drug, they want kind of low stakes research to make sure that whatever research they're running is going to work. So you're given a lot of money, but not necessarily a lot of flexibility. Where in academia, it's kind of the other way around. You're not given very much money, but you're kind of free to do whatever you want, which is obviously kind of preferable.
hmm,
So yeah, I kind of sway between the two. I think they both kind of have their own swings and roundabouts.
yeah.
And I haven't quite settled on an option yet, I don't think.
That makes sense. Well, who has really?
Yeah.
So I can definitely appreciate that position. Um, yeah, I mean, I've heard quite a bit about industry, and I mean, there is a bit of a reputation that sometimes pharmaceutical companies get, but I personally think that there's an immense amount of like beautiful research going on in there. I've recently heard that there are new drugs which are designed to wean people off of addictive substances
Mm,
like heroin, for example, which have been found to be significantly less addictive themselves.
mm.
So that seems like a really like that's what we should be doing. Honestly, so I can definitely appreciate the value of that. In your case, do you think that if you went into industry, uh, would you be focused more on these kinds of applications, or still something focused around your present research?
Um, I think that will be a question for the end of my PhD to
Mm-hmm.
see whether or not I still love the vascular system or whether I hate it.
Mm-hmm.
Right now, it's still very much a love hate relationship. It's super, super interesting, but maybe when my analysis isn't running quite right, I don't love it as much.
Mm-hmm.
But I've stuck with it now for 4 years, and I can't really see myself changing because there are just so many different options you can take. You can look at how different risk factors kind of contribute to different pathology within the brain, how different cognitive domains are effective, how different cognitive domains are affected, um, and then how this changes depending on what point of your life you're at, and all these there's so many different approaches that you can take. So I can't really see myself straying too far from that.
That makes sense. I've heard that throughout when you're doing a graduate degree, um, your purpose is kind of to specialize, hyper specialize almost, to the extent where you become world expert at something very, very
Yeah.
small. And then you have the chance as a person who has graduated already to then re-diversify. So is that something that you've experienced yourself, or do you think that you've had the chance to um, really explore a couple of different subfields?
Yeah, I think definitely my field is very specific.
Hmm.
But thankfully the lab that I'm part of, I mentioned this to Hugo earlier, I'm part of what's called the MISC lab. And it's called the MISC lab because we kind of represent miscellaneous disciplines, but also it stands for modeling, imaging, stress, and cognition. So different people in the lab kind of specialize in different components of MISC. So being part of a lab, I'm able to experience other people's research. So we've got quite a lot of stress researchers, so I'm able to have a bit of an understanding as to how stress responses occur,
Yeah.
how early life stresses influence the brain. And then we have a couple of other people that specialize in multiple sclerosis, so I also know a little bit about that. So I think being part of a lab has been really critical in ensuring that I'm not hyper specialist in just my topic, but also share some expertise in other topics as
Yes.
well.
Yeah, that totally makes sense. So, in other words, because you have the community of people who do slightly different
Yeah.
things, you actually do get the chance to talk with them about, okay, now I have Megan maybe for stress and other people for other subfields within my broader field. And so that gives you a bit of a opportunity to understand this industry at a broader level than you would have otherwise.
Yeah, not even just understand, but also collaborate. So
Mm,
we have a visiting master's student in our lab at the minute from Amsterdam, and she's interested in multiple sclerosis. And we are both going to be using the disconnectome approach because when you have multiple sclerosis, you also have these white matter lesions, which impact cognition in different ways. So we're both going to be using the disconnectome analysis. It's going to be really interesting to see whether our results are similar, whether they're different, whether cognition is impacted the same across both cerebrovascular burden and multiple sclerosis. So not just an opportunity to have more understanding, but also to collaborate with other researchers from different disciplines.
mm, perfect. So that sounds like a real way to get out of that narrowing problem that's many PhD students seem to have. And so I'm also curious, considering that you do get the chance to uh, see how different subfields work and even to collaborate with them. Do you think that informs and helps improve the kind of research that you're able to do?
I think definitely because I think when you come to do your analysis, it's really easy to just stick with what you know.
Mm,
So let's say you've always ever done ANOVAs, it's really easy to just stick with that and not learn other different analysis techniques, or even not know what else is out there.
right.
So I think being part of this lab, especially when there's such a heavy modeling component, You see how people are using similar data but differently, and they have different approaches to analyzing this data. And I think that's really been useful in shaping quite a lot of my research.
Yeah.
One of the analyses that I did in my biobank paper is what's called a principal component analysis, and that's essentially a data reduction technique. And that was inspired by someone else that was in the lab. So it's really been part of this lab has shaped quite a lot of my research, which has been nice.
Yeah, yeah. Personally, I remember being a part of some pretty innovative teams when I was uh, working as a software engineer. And to me, what was always amazing when I saw the kind of work that we could do was, you can contribute one innovation, but then four other people contribute their own one innovation, and collectively you have five. So it's like you multiply your collective potential fivefold with five people who are really dedicated to what they're doing. So personally, I find it to be really like your experience to be really resonant with me, and I'm sure that for people who are looking to do science themselves and are looking to understand a bit more about their field, it's a great idea to really try to connect with as many researchers and try to connect with, especially have really deep relationships with certain researchers around you to really get that sense of, "Oh, I actually can learn something from them in addition to giving something to them." And so that's what um, brings me to another question, which is I recall that you mentioned you go to conferences every
Mhm.
now and again. And so that seems to be another illustration of that idea that you're getting to interact with other scientists. So could you describe that to me a little bit?
Yeah, so the type of conferences that you'll attend as a graduate student will really depend on your specialty and what your focus is. There are so many different psychology conferences. There's the British Neuropsychological Society conference. There's the British Neuroscience conference. There's so many different ones, and I'm sure outside England as well. But the conference that we predominantly go to in our lab is called OHBM, which stands for the Organisation for Human Brain Mapping, and that's quite a heavily neuroimaging focused conference.
Yeah.
But people come at it from very different perspectives. So you can have quite a lot of methods based researchers who are interested in fine tuning analysis techniques or different kind of scanning paradigms. And then you have quite a lot of clinicians who come at it from a clinical perspective. So how can we use neuroimaging in clinical work? And then you also have researchers like myself that just use imaging within our academic research. Um, and OHBM is an incredibly big conference, so I think there can be up to 3,000 people that go every year.
Yeah.
And it's in a different place every year. So last year it was in Canada, this year it's going to be in Korea. And the past 2 years, I've had experience presenting my research as a poster at these conferences, which can be super overwhelming
Yeah.
because there are so many posters and there are so many talks, and the conference normally runs from 8 till 6 every day for 5 days, and
Yeah.
there's social events going on afterwards. So it's a really, really tiring experience, but it's such a good opportunity to meet other researchers. And I found the best way to do things is to look at the schedule before you go and put a pin in things that you definitely want to see and posters that you want to see. And make sure you go to that poster during the poster times and speak to the researcher and build that connection, because if you're chatting to them, the likelihood is that they share a similar research interest to you.
Mhm.
Um, so making the effort to go and speak to them and building your connections will really help you find a postdoc position after your PhD.
Mm.
And it means you'll also keep an eye out for their papers, and they'll keep an eye out for your papers as well. But like you say, it's just such a good opportunity to kind of build your sense of community. At OHBM, I also have a committee position as the career and mentoring manager, which has been really interesting. I've held that for the past year, and then I'll hold it again next year. And that basically involves organising one of the symposiums at the conference, and I also organise something along with my other mentoring managers called the Lunch with Mentors program,
Mhm.
where we basically select mentors that are quite advanced in terms of the career. So often professors. I think we normally pick about 15, and then we give postdocs and PhD students the chance to sign up, and they basically get to have a lunch with their mentor and chat about research opportunities, career progression, all that different stuff, which has been really, really, really rewarding. And I'm looking forward to seeing it again this year.
Yeah. That seems like something that could really bear fruit quite a bit because you're connecting the people who want to do some work with people who have the ability to give them that. So that seems like a really great opportunity for students who or students, well, graduates who are interested in something like that. And I was curious because you mentioned the poster that you've been working on, and what is it exactly about? And generally, because I know that that's a activity that many graduate students have to do or choose to do as graduates, and it's the making of these displays of your research.
Mhm.
So, in your case, what would that be? And also, how do you go about making one?
Yeah, so the poster that I've made to present this year is the biobank work that I've kind of
Hmm.
spoken about earlier. Um, and how I make it was a little bit easier for me this year because I have already written up my biobank paper. So it's just a case of kind of condensing down the information I have and putting it into a poster. But I think top tips for posters is to make it as visual as you possibly can.
Yeah.
Include lots of nice graphs, lots of nice figures. Don't skip the content. Make sure you still have the important bits there. I think it's always helpful to have a solid introduction and a solid conclusion section. And I think the methodology and the results section can generally be told with figures and with really nice graphs and really nice brain pictures or whatever it is you have. So I think try and make it as gorgeous and beautiful as you can.
Hmm.
Um, I normally make my posters in a website called Canva, which is really great, and they
Ah, yeah.
offer really amazing templates and things like that. But I know there are lots of others available online.
Yeah, yeah. I remember using Canva myself, so
Yeah.
definitely can attest to it being a useful tool. Well, thank you very much for this interview, Katie. This was really, really great, and I think a lot of people will get the chance to hear about what you've been saying and hopefully to maybe reach out themselves.
Yeah, feel
So,
free.
yeah. So, let's do this again sometime. Thank you very much.
Thank you.